Hello, and welcome to this introduction to our tool, Bulk Analyzer. My name is Eleanor Williams, and I'm a PhD student in the Core Bioinformatics group led by Dr. Irina Moriani at the Cambridge Stem Cell Institute. Today, I'm giving a brief overview of Bulk Analyzer, which is an accessible, interactive pipeline for analyzing and sharing bulk sequencing results, with functional approaches focused on gene regulatory networks for integrating multiple modalities and datasets at the bulk level. Bulk Analyzer creates easy-to-use Shiny apps to increase openness and reproducibility of research and increase connection with the community. In recent years, bulk sequencing data has become very widely used for hypothesis generation across a spectrum of biological questions, and a huge range of tools have been created for analyzing the data. However, combining these different methods often requires dedicated bioinformatics support or often expensive and inflexible commercial solutions to streamline the process. We created Bulk Analyzer as an open source and accessible approach to combine multiple analysis options, including differential expression, gene set enrichment, and pattern identification, as well as pre-processing and quality control steps. So here is the workflow of the tool. Bulk Analyzer is an R package which is specifically designed to be user-friendly and can create an interactive Shiny app with only 2 lines of code. The input is a raw expression matrix which could be obtained by standard quantification using, for example, feature counts, plus a metadata table describing the samples. Denoising using noisy R and normalization using a choice of methods, including quantile normalization, TMM, and DESeq2, among others, are all handled by the pre-process expression matrix function. From there, you can use the other central function of the package, generate Shiny app, to create an app. This app performs all of the analysis shown in the workflow on the left and can either be kept internally or deployed in public so that the wider community can interact with your data and gain insights of their own without completely reprocessing it. I'll now show an example to showcase some features of the app. So you can firstly select to focus on all samples or only a subset of samples. So if you wanted to look at one condition at a time, or if you wanted to exclude individual samples, which you've identified as outliers. You can then perform quality checks, such as a heat map showing the Jacquard similarity index on the top genes, and principal component analysis. And in both cases, you can alter the genes that are used, for example, and change how the plot looks. There are then MA plots, density plots, and you can also query the expression of individual genes of interest and see how these change across all of your samples. There are then panels for standard analysis options like differential expression and gene set enrichment, and you have lots of user-defined parameters to change how this analysis is run. You get an interactive table giving the differential expression results, and you can highlight some genes of interest, which will then be emphasized in volcano plots, MA plots, and heat maps. You can also compare between differential expression calls with cross plots, and all of these plots are clickable, so you can find out what the genes that are coming up are. And you can infer expression patterns across a sequence of samples, classifying genes into up and down patterns. Bulk Analyzer also enables the inference of gene regulatory networks using Gene3. So you can choose to run this on all samples or only on a set of conditions in order to compare between GRNs across groups of samples to understand the GRN dynamics, both in terms of changes in topology and strength of interaction. You can pick one or multiple target genes, and then you can increase the number of connections shown and start to explore the data and the similarities and differences between the different GRNs that come out. From there, there's a host of ways to enhance the GRN inference within Bulk Analyzer. Firstly, by adding enriched terms from the gene set enrichment analysis that are associated with multiple genes in the GRN, and by adding custom external annotations. You can integrate multiple modalities through cis-regulatory interactions, such as integrating ChIP-seq and RNA-seq data, and trans-regulatory interactions, like combining messenger RNA-seq and microRNA data. By integrating multiple modalities, you can create a combined network to enhance understanding of how your whole dataset fits together. Also, Bulk Analyzer is agnostic to the type of count data used as input. And while one Bulk Analyzer app can contain a whole set of tabs for several different modalities, as well as GRN tabs integrating them together. So this was only a brief introduction to Bulk Analyzer, but if you're interested to find out more, have a look at the QR codes shown here for the Bulk Analyzer paper published in Briefings in Bioinformatics, the package on GitHub where there's lots of documentation, and an example app to play around with. Thanks for listening. I hope to see you in Lyon.